Welcome to Prospective Doctors MCAT Podcast, brought to you by Med School Coach. Each week, Sam breaks down the highest yield MCAT topics so you can score as high as possible on test day.
Hello and welcome to Prospective Doctors MCAT Basics with your host Sam Smith. The goal of this podcast is to cover the highest yield topics on the MCAT and provide you with some sort of insight into the questions that the MCAT really likes to ask. This podcast is going to cover the thing that you see out of, which is the eye. And I'll first start by breaking down the eye in terms of its anatomy and physiology. I'll talk about the components of the eye and their function as well. And then I'll get into rods and cones, spend a bit of time there, talk about things like the phototransduction pathway, and then I'll get into perception. And in terms of perception, I'll talk about visual field processing, I'll talk about feature detection, and I will talk about parallel processing. And this material will show up in one out of the 4 MCAT sections. That's going to be the Psych-Soc section. And I've always found this stuff pretty interesting. I mean, it's kind of amazing to me that just based on the way that light enters the eye and is focused on the back of the retina, we can see all these objects around us and colors and everything just perfectly crystal clear. So, I hope you enjoy this podcast. I hope you learn some good things out of it, and good luck as you study for the MCAT. All right, so the first thing I want to talk about here are the different parts of the eye. And so what I'm going to do is I'm going to essentially introduce the different parts of the eye going from the outside in. And then while I'm doing that, I'll kind of just briefly talk about the function of each of these different parts. All right, so pretend that you're looking up at the sun and there's a ray of light that just traveled for 8 minutes and 20 seconds from the sun all the way down to the earth, and it enters your eye. And so the first part of your eye that light ray is going to hit is called the cornea, which is the clear front window of the eye. And essentially, the cornea acts to focus the light that enters the eye. And it accounts for approximately 75% of the eye's focusing power. And this is something that I kind of didn't know. I really just assumed that the lens of the eye was like the main focuser of the eye. Well, it just so happens that that's not necessarily true. It's actually the cornea that focuses the majority of the light that enters your eye. And on top of focusing the light, the cornea also acts to protect deeper and more important parts of the eye. You know, stuff like the iris, the retina, the pupil. It kind of serves as this outer protective layer that protects those inner layers that have kind of more of an important function, I guess, than the cornea. The next part of the eye that I want to talk about is called the iris. And this is the colored part of the eye that helps regulate the amount of light that enters the eye. Essentially, what the iris does is it kind of acts like blinds for the eye. So it controls either how wide the pupil is or how narrow the pupil is. And eventually it controls then the amount of light that is actually able to enter the eye. And so obviously then the next part of the eye that I'm going to talk about is called the pupil. And this is the dark spot in the center of the iris that, again, lets light into the eye. And what's interesting is that the pupil appears to be black because all the light that enters through the pupil is actually absorbed by the eye at the retina, and therefore no light is emitted out of the pupil, and so the pupil appears to be black. So that's kind of interesting. Again, the pupil is essentially the window of the eye that lets light into the eye, while the iris are like the blinds of the eye that control how much light gets into the eye. And don't forget that the iris is responsible for what most people would consider the most important part of the eye, which is the color of the eye. So for example, I have red eyes, and that is due to my red iris. It's also important to note that the iris and the pupil have this reflex known as the pupillary light reflex. And essentially what this reflex does is that under conditions of lots of light exposure, the pupil will constrict, therefore letting through less light into the eye. And then when you're in conditions of low light exposure or when it's dark out, your pupil will dilate and therefore let in more light, so then you can see a little bit better in the dark and you don't just destroy your eye in the really bright conditions. You know, maybe you're looking straight at the sun or something like that. All right, so as we move deeper into the eye, we come upon what is known as the ciliary body. And this is a structure that contains a very important muscle, which helps focus the lens. Essentially what happens is that when the ciliary muscle contracts, the lens either becomes more convex, i.e. the curvature increases, or it becomes more flat, in which case the curvature would decrease. And when it becomes more convex, it actually helps the eye focus on closer objects, whereas when it flattens, it helps the eye focus on further away objects. So if we were to compare our eye to a camera, the ciliary body would be essentially the zoom, right? That helps us see close objects clear, or if we want to see further objects away, we can focus on those. And this is due to the contraction of the ciliary muscle. And this process of either flattening the lens or making the lens more convex is called accommodation. And that brings me to the next part of the eye, which is the lens, which is kind of the functional part of the eye. Maybe the most interesting. From a physics perspective, I think it's probably the most interesting. But anyways, what the lens does is it takes light that has been kind of pre-focused, I guess, by the cornea, and it further focuses that light right directly to the back of the eye in a place known as the retina. And the retina contains these cells that are able to sense light and then send that signal to your brain so that you can see an image. And I'll get into these cells that are able to sense light. These are called rods and cones. It's kind of interesting that this lens in our eye is actually made up of a protein called crystalline. It's a water-soluble protein, and essentially this protein increases the refractory index of the lens, which if you remember back to physics, that means that it makes the light slow down as it enters the lens and therefore refract at some angle, and eventually this leads to the light being focused at the retina. And it's important to remember that in terms of focusing power, the cornea actually has the majority of the focusing power in the eye, that being about 75%, whereas the lens has a minority of the focusing power, which is about 25%. So that's something I didn't know. I mentioned it a little bit earlier, but just something that I think is important not to forget. All right, the next part of the eye that I want to talk about is called the sclera. And the sclera is the white outer coating of the eye that surrounds the iris. It's the quote-unquote white of the eye. And what it does is it provides structural integrity and support for the eye and is primarily made of collagen, which obviously is also found in a lot of other connective tissues in the body, in your joints, ligaments, even in bone. So again, the sclera is the white part of your eye that is made up of collagen and serves a role in providing structure and support for your eye. All right, as we continue to travel through the eye, we reach this clear, gelatinous, kind of nasty substance that fills the central cavity of the eye. And this substance is called the vitreous humor, which is not to be confused with aqueous humor, which is not to be confused with poolside stand-up comedy. But anyways, the vitreous humor is this gelatinous substance that fills the inner cavity of the eye, and it gives the eye its structure. So it's kind of like water to a water balloon, right? If you had just this deflated balloon, it wouldn't be circular. It wouldn't be the shape of an eyeball. So therefore, the vitreous humor then provides the eye with its structure and shape. And fun pathology fact of the day for you, the vitreous humor is preferred to blood for most post-mortem biochemistry analyses, since it is thought to be far less susceptible to autolytic change after death, which is just basically to say that vitreous humor is more preserved after death than the blood. I guess that's kind of how I see it. So as we make our way through this vitreous humor, eventually we reach the back of the eye, which contains a very thin layer of cells known as the retina. And the retina is really, really important for the function of the eye. And I know I've said this probably about every single part of the eye so far, but the retina is really important because it has light-sensing cells called rods and cones that are able to actually detect light, and in the case of cones, are able to actually detect different wavelengths of light and therefore different colors, and then transmit that information to the brain through the optic nerve. And in a minute or so, I'll do a deeper dive into these rod and cone cells and talk a little bit about how exactly they're able to sense light, what occurs in the cell in order for it to transmit signals to the brain. But before I do that, I also want you to know that the retina has a few different important parts to know. The first of which is called the macula, which is at the center of the retina and contains a very high density of these light-sensitive cells, which allows us to see very clear, fine details. So I kind of like to think of the macula as like the functional center of the retina. And in the very center of the macula is a part of the eye known as the fovea, which has the highest concentration of cones of the retina. So again, this part of the retina has a lot to do with being able to see very fine details. So, you use your fovea to be able to clearly read books, play video games, see these really very, very small details. And so overall, you have a retina that's at the back of your eye. It's a very thin layer of cells. And in the middle of the retina is the macula, which contains a high density of rods and cones. And then in the middle of the macula, you have what's known as the fovea that has a very, very high density of cones only. So, you could almost imagine it like a target, like the outer ring, then inner. The outer being the retina, macula, then fovea. And the last structure in the eye that I want to mention is called the optic nerve. And what it does is it connects the eye to the brain and basically carries electrical impulses that are formed by these light-sensing cells in the eye up to the brain, and then the brain is able to perceive what you're seeing and kind of put it in context for whatever situation you're in. So those are some of the basic structures in the eye to know for the MCAT. The next thing I want to talk about is how we turn light that enters into our eye into signals that can then be sent to the brain in order for us to know what we're looking at and perceive things that are in our field of vision. All right, so as I said earlier, light enters the eye through the cornea. It gets focused through the cornea and then passes through the lens, which does the final bit of focusing. And the lens specifically focuses the light at the back of the eye in a place called the retina. And the retina has these cells that are able to actually detect the light. And these cells are known as, number one, rods, and number two, cones. So let's first talk about the difference between rods and cones. So rods are concentrated on the outer edge of the retina, and they have very limited ability to detect light. However, though, they are very sensitive to light, and so they're responsible for almost all of our night vision, even though obviously we don't have that good of night vision. And this gets to the fact that it is hard for us to make out colors in the dark, right? Go out in the dark at night and try to make out what colors the cars are that are passing your house or whatever. It's kind of hard to do, and that has to do with the fact that our rod cells, which are very sensitive to light, aren't able to really detect colors. And in terms of a rate of response, rod cells are much slower to respond to light than cones are. And therefore, rods have pretty low spatial acuity. And what I mean by that is that they're unable to produce really sharp images that you see. And so when you think about which type of cells, either rods or cones, are able to produce these sharp images, think cones, don't think rods. And that all has to do with the fact that rods respond much slower to light than do cones. Cones are able to actually respond a lot quicker and respond a lot quicker to changes in light. So that's a brief kind of intro to rods. Cones, on the other hand, are capable of sensing color. So when you think of color vision, think cones. And that's pretty easy to remember, right? Just remember the C for cone is equal to color vision, CC. So it's pretty easy to remember. And essentially this works because you have three different types of cones that are able to sense different wavelengths of light. So you'll have a shortwave cone or an S cone that is sensitive to, you guessed it, short wavelengths of light. Then you have medium waves or M cones that are sensitive to medium wavelengths of light. And then you have long wave or L cones that are sensitive to longer wavelengths of light. And in contrast to rods, cones tend to populate areas closer to the center of the retina. So they're mostly found in the macula, and they're exclusively found in the fovea, as I already said. And as I was kind of talking about when I talked about rods, cones are a lot less sensitive to light. So in darker conditions, they might not be able to sense light as well as rods can. And Cones are also responsible for our ability to see really sharp, clear images. So, reading, as I said, video games, stuff like that before. Cones are really good at responding quickly to changes in light, and that really helps us be able to see sharp, clear, crisp images. So let's talk now about how these cells are actually able to detect light. And I'm going to first talk about this in terms of rods, and then you can kind of broadly apply this to how cones work too, essentially, but with a few caveats. So I'll first talk about this in terms of rods, and then I'll tell you what the caveats are in terms of cones. So rods use this protein called rhodopsin, and through a pathway known as the phototransduction pathway, which is very cool, they are able to sense light. And so let's talk about how this works. So rhodopsin is a GPCR, which is a G protein coupled receptor, which is a membrane protein. And this membrane protein is found obviously in the cell membrane of rod cells, because we're talking about rods. And it consists of two parts. It consists of a protein molecule that's called scotopsin and a covalently bound cofactor called retinal. And what happens in the phototransduction pathway is that light hits rhodopsin, and specifically retinal, and causes retinal to undergo an isomerization. It goes from an 11-cis retinol configuration to a all-trans retinol configuration. So it isomerizes, and it's very important to know that it takes a photon of a very specific wavelength to make this happen, which is about 500 nanometers for a rod. And I'll get into why this is important, but just kind of hold in the back of your mind right now that this is how cones are able to detect different wavelengths of light. And so, again, we're talking about rods, though. And so what this does then is once retinal isomerizes, it sets off this GPCR, which leads to hyperpolarization of the rod cell, which is kind of weird, right? Why would that hyperpolarize the rod cell? Wouldn't it depolarize the rod cell in order to send an electrical signal to, up the optic nerve to the brain? Well, photoreceptor cells are weird in that they depolarize in response to the absence of a stimuli. And so essentially this is what's going on. In dark conditions, the optic nerves are inhibited by rods and cones. These rods and cones actually release glutamate, which is an inhibitory neurotransmitter. And so these nerves are just inhibited. But boom, as soon as you get into light, these cells are then hyperpolarized, which decreases the glutamate released from these rods and cones, and therefore leads to the excitation of nerve cells. So it's this kind of weird pathway, but just kind of understand overall that light causes a conformational change in retinal that activates the GPCR pathway in rhodopsin, causing the cell to hyperpolarize, which then transduces a signal through the optic nerve to the brain. So that's how rods work. What about cones? So cones have a different protein in them than rods. This protein is called photopsin, which is very similar to rhodopsin, but it's in cones. So remember that. Rhodopsin, rods, you can match the R's together, and then cones have photopsins. And like I said earlier, there are three different types of cones, right? And they are either the S cone, which is a shortwave cone, the M cone, which is a medium wave cone, or the L cone, which is a long wave cone. And so essentially the difference in these three cones is a few amino acids that make them sensitive to different wavelengths of light. So, there might be a threonine in an S cone, whereas there might be a tyrosine at that same point in a M cone, which now makes it more sensitive to a longer wavelength of light. And I just made that up, but it's something like that. There's a very few changes in amino acids that make these three different cones most sensitive to three different wavelengths of light. And just to get a little physicsy, the S cone is sensitive to wavelengths between 420 and 440 nanometers. The M cones are most sensitive to wavelengths between 535 nanometers and 545 nanometers. And then L cones are most sensitive to wavelengths between 565 and 580 nanometers. And I sure as hell would not memorize those numbers. It's just something kind of interesting, something to think about, something to start thinking about how the MCAT is going to ask you questions. They might ask you to do some calculation where you got to calculate the energy of a photon that is absorbed by an S cone that it absorbs within this range of wavelength, blah, 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 blah. Some BS like that. So anyways, just be cognizant that there are 3 different types of cones, and they absorb light differently based on the amino acid makeup of their photopsin protein. Alrighty, so I hope that gives you a little bit of a look into the phototransduction pathway, something that you definitely could see on the MCAT. The next thing that I want to talk about here are a few different common maladies of the eye. And I've actually seen these come up in the physics sections of the MCAT, but you might see them in the psych soc too, you never know. And what I've done is I've broken these maladies down into 2 different categories. The first are refractive errors, and the second are age-related eye diseases. So let's first talk about refractive errors of the eye. And this is the more physicsy of the 2 malady categories. And so the first 2 that I want to talk about are called myopia and hyperopia. Myopia, which is also called nearsightedness, is when the lens focuses light to a point in front of the retina. So you go back to your physics class. Do you remember the thin lens diagrams? If not, go listen to my podcast on it. But essentially what's going on is that the curvature of the lens is too great, and therefore you are focusing the light to some point in space a little bit before the retina, and that is actually going to make objects that are close to you appear blurry, but objects that are near you appear clear. So I guess it's important to remember that nearsightedness is just being able to see near objects clearly, but then it's hard for you to see far away objects, or far away objects appear blurry. And I'm getting a quick update in my ear from my producer. Okay, so I guess what I said was not necessarily all that true. Apparently, this disease has to do more with the shape of the eyeball. So although it can happen where you have a misshapen lens, what occurs more often is that you have a misshapen eyeball where your eye is actually lengthened a bit. And so essentially what happens is your lens is actually focusing light to where the back of your eye should be, but your eye is misshapen and it's kind of lengthened out, and so that is not actually where the back of your eye is. On the other hand, hyperopia is the exact opposite condition in which a person has farsightedness. And in this case, the lens of the eye focuses the light to a point behind the retina, like into the brain essentially. Obviously, this light can't actually make it into the brain though, because it just hits the back of the retina. But of course, this is unfocused and leads to what's known as farsightedness. And in this case, you can clearly see objects that are far away, but you cannot clearly see objects that are close to you. And both of these diseases can be fixed through the use of glasses. There is a more serious malady of the eye that is a refractive error, and that is known as an astigmatism. And it is due to an irregularly shaped cornea or lens that prevents light from focusing properly on the retina. And essentially what's going on in this case is that the eye shape causes the light to refract in weird ways into the eye, resulting in things looking blurry to you. And in this case, it's a little bit different than myopia and hyperopia in that your lens might be correctly focusing light at the back of the eye, but in this case, you might have multiple focal points just because the curvature of your eye is weird, or the curvature of your lens is weird, or maybe you've had some damage due to an injury, whatever it may be. Now you have a bunch of little focal points at the back of your eye, and your brain is like, what the f***? So these are the main refractive errors of the eye to know. Again, I've seen these come up in the physics chem section of the MCAT, not just the psych soc. I want to get in now to the age-related diseases of the eye. The first I want to talk about is called age-related macular degeneration. And this is caused by degradation to the part of the eye that is able to detect light. Again, this is the retina, or the functional part of the eye. And this usually occurs due to damage or cell death of rods and cones that are in the macula. And there are 2 different types of macular degeneration. One is called wet macular degeneration, and the second is called dry macular degeneration. But unless you really want to be an ophthalmologist or something like that, you really don't need to know the difference between these two. So in terms of the MCAT, this is out of the scope, so I'm just going to kind of skip over that. The next age-related eye disease you might see is called glaucoma. And glaucoma is a group of eye diseases that result from damage to the optic nerve. And this damage to the optic nerve is typically done by abnormally high pressures within the eye. And so to summarize these age-related conditions within the eye, you have age-related macular degeneration, which is death or damage to rods and cones that make up the macula. And then you have what's known as glaucoma, which is a group of diseases that result from damage to the optic nerve, which is obviously very, very important for sending signals from the eye to the brain to let your brain know, hey, I'm looking at my phone right now, or hey, I'm scrolling through my Instagram right now, or whatever you're doing. All right, so that's a little bit of a dive into the eye, some physiology of the eye, and then also a little bit of pathophysiology of the eye. The next thing I want to talk about is perception. And this is going to be a lot more of the psych-soc stuff, but this stuff is also very important for the MCAT. All right, in terms of perception, I want to talk about three different things. The first is visual field processing. How do these nerves look as they go up to the brain? The short answer is they do a little crisscross thing. The second thing I want to talk about is binocular versus monocular cues. Then the last thing I want to talk about is feature detection. And within feature detection, I'll get into a few different things, being Gestalt principles, parallel processing, and the Parvo versus Magno pathways. All right, so let's get into visual field processing.
Thanks for checking out the Prospective Doctor MCAT Basics podcast. Sam's doing a killer job taking you through the most important MCAT topics, but what if you need a little extra help? How does a 5, 10, or even 15-point increase in your score sound? Imagine how your chances at admission could increase. Med School Coach's MCAT tutoring can get you there. With the most rigorous selection process of any tutoring company, we see amazing results. We deconstruct each student, find a plan that is going to work, and help execute it. That's why our students add an average 12 points to their score. Completely physician-run and operated, and focusing on nothing but medicine. It's no wonder over 10,000 past students have trusted Med School Coach to get them through the MCAT and into medical school. Check out medschoolcoach.com today and mention code PODCAST for 5% off.
All right, so let's get into visual field processing. All right, so let's get into visual field processing. As I said in the intro, the optic nerves from each of the eyes do this weird crossover thing. Shout out Allen Iverson. And overall what happens is that the right side of the visual field is processed on the left side of the brain, and the left side of the visual field is processed on the right side of the brain. So let's talk about how this occurs. And as always, it would really help to have a picture in front of you, or go look at this later, but you're going to hear a lot of rights and a lot of lefts, and it's going to be kind of a mental puzzle to be able to keep this all together. But let's go ahead anyways. So imagine taking each of the eyes and cutting it in half lengthwise. So you're cutting from the optic nerve to the front of the eye. And this is going to split the retina in two. And essentially, each side of the retina processes a different part of the visual field for each of the eyes. In other words, the retina from one of these slices is gonna process part of the visual field from one side of your body, and the retina from the other side of this half eye that you've cut in half is going to process the visual field from the other side of your body. So it goes like this. In the left eye, the left side of the retina processes the right side of the vision field, and then the right side of the retina processes the left side of the vision field. And then in the right eye, the right side of the retina processes the left visual field, and the left side processes the right vision field. So it's a bunch of crisscrosses going each way. A little bit confusing. Again, look at a picture if you're a little bit confused. And this is a little bit of a simplification. I'm sure that there's some cells, on the left side of the retina that process the left side of your vision field. But this is just kind of a general pattern to understand. And so what I want to talk about next is what this means for the nerves that travel from these different parts of the eye. So let's take, for example, the right side of the right retina. So as I said, that processes the left side of the vision field. So what does that mean for the nerve that runs from that part of the retina to the brain? Well, as I said before, overall, the right side of the brain processes the left side of the visual field. So those cells that are on the right side of the right retina that are processing the left vision field don't have to cross over to the left side of the brain. Instead, that nerve runs all the way from the eye to the brain without crossing over to the left side. However, if you are the left side of the right eye, well, then you are processing the right side of the vision field. So then that nerve has to cross over to the left side of the brain in order for it to be processed, because again, the right side of the vision field is processed by the left side of the brain. And that is a kind of a verbal description of how this process works. Of course, that might not be all that helpful. It's kind of sounds like a nerdy electric slide. There's a bunch of right, lefts, turn, shuffle, slide, whatever. So that might not be helpful. If it isn't, go look up a picture and you'll kind of see what I'm talking about. But just know it's important to understand how these different crossovers work. And really overall, just understand that the right side of the vision field processed by the left side of the brain, and the left side of the vision field processed by the right side of the brain. All right, let's talk next about monocular versus binocular cues. So monocular cues are visual cues that we only need one eye to be able to recognize. So this is things like relative size, shape, texture, right? You only need one eye to be able to tell the shape of an object, or one eye to be able to tell the texture, right? Right now I'm looking at a bowl of oatmeal. I can see that bowl of oatmeal is thick with one eye. So that's just kind of a little example of that. There's another one that you might see come up on the MCAT that's a monocular cue called motion parallax. And I know what you're thinking right now. You're thinking motion parallax? Is that not a Dr. Seuss book? Well, it's not, but what I'm going to do is I'm going to try to describe it using Dr. Seuss language, and we'll see if you're able to tell what I'm talking about. Here we go. Far objects move slow, like a distant river, yo. Up close, it might whiz by, but take a few steps back, and it does not lead the pack. I kind of hate myself for doing that, but if you're on the MCAT and you see this term come up, I hope it reminds you of Dr. Seuss, and then you remember the definition of motion parallax, which is that we are able to perceive objects being far away when they're moving slow. So, if a car is really far away, it's moving slow, or it appears to be moving slow, or we perceive it to be moving slow. However, if a car is very close to us, then it moves really fast and we perceive it as moving really fast. And therefore we say, okay, that car is pretty close to us. So that is motion parallax. And I hope that it comes up on the MCAT and you chuckle a little bit because you remember Dr. Seuss. And further, motion parallax is a monocular cue. We only need one eye to be able to do it. On the other hand, binocular cues are visual cues that we need two eyes to be able to recognize. The most prominent example of a binocular cue is retinal disparity, which refers to the slightly differing views that we get from each of the eyes, right? So each of the eyes are spaced a little bit apart, and so each of them gets a slightly different view of an object. And essentially, this enables us to perceive the relative distance of objects in our visual field. It kind of gives us almost like a 3D vision, or what's known as depth perception. But beyond just these examples that I'm giving, know how to kind of apply the concepts of monocular cues, which require only one eye, and binocular cues, which would require both eyes. For example, I could see the MCAT giving you some psych-soc passage about a study that's been done on children, and let's say that, they split children up into two groups. Half of these children see a really violent video, and then they're placed in a room with this doll. And then the scientists are trying to measure, their level of aggression through their interaction with this doll. Maybe they're punching the doll, maybe they're kicking the doll, maybe they're kissing the doll. And those are all kind of giveaways of their attitude. And so maybe it asks you something like, this child that was seen punching this doll, does this child need monocular vision or binocular vision to do this? In which case, me personally, I would answer binocular vision, because you need depth perception to be able to land a punch where you want to. And that's kind of an off-the-cuff example, but just know these two monocular and binocular cues and how to apply them. All right, the next thing I want to talk about is visual feature detection, and I'll start by introducing the Gestalt principles. And just a quick healthcare aside, I've actually heard the word Gestalt come up when I've shadowed ICU doctors. And so one time I was shadowing and this doc goes, and my Gestalt on this case is blah, 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 blah. And I was like, wait, is he talking about like vision and like Gestalt principles? And I found out that in terms of healthcare, Gestalt can refer to a overall coherent theory that a physician has come up with throughout years of practice. So maybe there's not direct evidence for whatever route of care that they want to take, but they've developed this all-encompassing theory called the Gestalt that they try to follow when they see a certain case, or maybe their overall practice theory, or rules they try to abide by as a physician. So kind of interesting. In terms of our visual perception, the Gestalt theory says that we perceive objects as more than just the sum of their parts. And this theory is laid out nicely in 6 principles called the Gestalt principles. And it really helps to go look at a picture of these. And I know I've said this throughout the podcast, but there's some nice pictures on just like Google Images that show each of these principles and like a real example of them. Anyways, the first law is called the law of closure, not to be confused with the law of closure. And the law of closure says that we automatically fill in gaps in images in order to create whole shapes. In other words, we really prefer shapes. And so if you look at like a circle that essentially is like a dotted line, we don't just perceive little lines kind of just chilling. We perceive that to be a circle. And so we prefer those shapes. The next law is called the law of proximity. And this says that we tend to group objects that are close to each other together. It's pretty simple. Then we have the third law, which is called the law of similarity. And that says that we group similar objects together. Then we have the law of continuation, which essentially says that if we have 2 lines that intersect, we follow the curvature of each line to say that, okay, this is made up of 2 individual lines. So let's say you have an X, like just the letter. We tend to perceive that as 2 lines crossing instead of, 4 individual lines with a center point, or, 2 V's or whatever. And the next law is called the law of symmetry, and it says that elements that are symmetrical to each other tend to be perceived as a unified group. So if you have a line of dots and a line of triangles, we tend to see those differently because they are each symmetrical to each other, the circles to the circles and the triangles to the triangles. And then the last law is called the figure ground principle. And it says that people instinctively perceive objects as either being in the foreground or in the background. And so that was a kind of a quick run through of the important Gestalt principles. What I recommend you do is go look up these images on Google and put them on a note cards and make sure you know each of these different Gestalt principles. All right, the last thing that I want to talk about in terms of perception is what's known as parallel processing. And so parallel processing is a part of our feature detection and visual processing systems, in that the brain is able to process many and multiple details about an object at once. So for instance, our brain can process color, motion, shape, depth, and a bunch of other variables separately and at the same time, aka in parallel. For example, say I'm sitting out on my front porch and I'm just people watching and people are walking by. Well, I can look at somebody who's walking by and I can process a bunch of information about them at one time. I can see that number 1, they're moving. Let's say that this person has blonde hair. And let's say that I am, listening to this person talk all at the same time. And I know that that's not visual, but we're able to process that at the same time as well. So what's happening is that there are different areas in the brain that are dealing with these different parts. So, we have a color center in the brain. We have what's known as the Magno pathway, which deals a lot with motion. And so we're able to kind of through these different channels process these different bits of information all at the same time to provide a coherent full image of what we're seeing. And this is hugely important, right? Because if we're able to only process one of these things at once, it would be a shit show. I'd be looking at something and then I'd have to close my eyes and go, okay, color, and then open them up and just see the color, then be like, okay, close them again. All right, motion, and then look and see how it's moving. And it would just be really difficult to tell what you're even looking at. And so our brain is able to use parallel processing through a few different ways. And I kind of talked about this a little bit just a second ago, but in terms of color, we have these different cones in our retina that are able to pick up different wavelengths of light. And then information regarding color is sent through these cone cells to the color center in the brain where it's processed. And then if something is in motion, it tends to go through what's known as the magnocellular visual pathway, or the Magno pathway. And this pathway is heavily involved in motion detection, but has very low spatial resolution. What I mean by spatial resolution is, very fine details it can't really see. On the other hand, we have this thing called the parvocellular visual pathway, or the Parvo pathway, and that is involved in helping us see very fine details and colors. And it can still sense motion as with the Magno pathway, but not as good as the Magno pathway. So I tend to think of it that the Parvo pathway helps us see fine details and colors, and the magnocellular visual pathway, or the Magno pathway, can help us see motion as well as color, but really doesn't help us see any of these fine details. And this is pretty easy for me to remember because I just remember magno motion. I group the M's together. All right, so that does it for the Eye podcast. As always, thanks for listening. If you like this podcast or feel that it's helpful for your MCAT studies, please go leave us a review on iTunes. It helps us reach more people. My next episode is going to be an interview where I'm talking about retaking the MCAT. So I'm going to get pretty deep into the weeds on retaking the MCAT. Who should retake the MCAT? How do you even improve your score? What if you're really bummed out about your score and screw this med school stuff? So hopefully some people find that helpful. I know a lot of people are getting their scores back now. So maybe you're on the edge and you're like, should I retake? Should I apply? What should I do? I hope that podcast will be helpful for those of you who are still questioning these things.
Thanks for listening to Prospective Doctor's MCAT podcast. If you're a pre-med, you'll want to check out prospectivedoctor.com for tons of free tools, articles, and more podcasts that cover your pre-med life. And if you need help on the MCAT or getting into med school, check out medschoolcoach.com for the experts.